Good morning, everyone. Apologies that I can't be there in person today, but for those of you that don't know me, my name's Jess Taylor and I'm the Marine Senior Advisor at Natural England covering Hampshire and Isle of Wight. And today I'll be talking about 3 pieces of work that Natural England have undertaken, either alone or in partnership with the Marine Biological Association, to help build the evidence base on non-native species in the Solent. So the first thing I'd like to talk about are our condition assessments. But why do we assess the condition of our sites? So firstly, there is a requirement under Article 17 of the Habitats Directive to report on the condition of our European sites. So these are our special areas of conservation and our special protection areas. But alongside this formal requirement, understanding the condition of our sites is vital for informing advice on management and also underpins all of our casework responses, as we have to understand the condition to understand what the effects of any development may be on that site. This works alongside some key policy drivers, for example, the new Environment Act target for 70% of our sites to be in favourable condition by 2042, with the rest in recovering condition, as we must understand the current condition and any reasons for unfavourable status to know what measures will be required to change this. So in 2016, Natural England began the process of assessing the condition of the SACs in the Solent, starting with Solent Maritime SAC, the boundary of which is shown on the slide. So what did the results of the condition assessment show? The 4 results of the condition assessment are available in the summary report, which can be found on the Solent Forum website, a link to which is included in this presentation. But what it identified was there were several reasons for the unfavourable status of Solent Maritime SAC, including high nutrient levels, as well as high levels of contaminants in both the water and the sediment, and the loss of extent of certain features like seagrass and salt marsh. But what about non-native species, the reason that we're all here today? So subtitled, it was really easy to note the high levels of non-native species because of the dominance of the slip Olympic Crepidula faunacata. However, our data in the intertidal was largely made up of unquantified point data from multiple sources, but it wasn't clear what this meant for the condition of the SAC, as you can obviously have non-native species present, but not find that is resulting in unfavourable condition. As part of the process, we therefore identified non-native species as a potential condition threat, in part because the conservation advice has a reduced target, and so there is a target set to try and minimise the number of non-native species in the site, but with a key note that states that further survey was needed to determine whether or not non-native species were impacting native populations and the features of the SAC. And so we needed to find a way to address this gap. To do this, in 2017, in partnership with John Bishop and Chris Wood of the Marine Biological Association, we applied for money from the European Maritime Fisheries Fund, EMFF for short, for a 2-year project to try and answer 2 key questions. Firstly, to try and address that condition assessment knowledge gap, we wanted to look at whether non-native species were causing unfavourable condition of Solent shores. And secondly, we wanted to look at what the influence of marinas and harbours as possible sources of non-native species, as these are often highlighted as a key entry point. Now, it should be made clear straight away that marinas and harbours are definitely not the only pathway for non-native species, and some of those other pathways will be covered in slides by APEM a little bit later. However, the topic was chosen as it is a key problem, it is a key known entry point, and we already had some data on the levels of non-native species within marinas in the Solent from work previously undertaken by the Marine Biological Association. So in order to answer these questions, firstly we had to identify our sites. So 14 clusters of 3 sites were chosen to provide comprehensive cover across the entire Solent, as you can see on the map. Each cluster was designed to be formed of 1 marina, 1 site near to the marina, known as the near shore site, and 1 site further away, or the far shore site. Now you can probably see from the map the distances between the marina and shore sites were not always the same, as shore sites had to be chosen very carefully based on things like access safety, making sure it was within the boundary of a designated site, but also making sure that the features of the SAC that we were trying to look at the condition of were actually present. However, all of the shore sites were chosen to ensure that they could not be influenced by another marina or harbour. And all were defined in advance. So all of the shores and all of the marinas were identified prior to all of the survey work happening. Surveys were undertaken at each of these 14 clusters over 2 years from May to July of 2018 and 2019. All survey work involved 3 people, which was John Bishop and Chris Wood from the MBA and myself, conducting a 1-hour time search at 14 marinas and 28 shores, as highlighted by the map on the previous slide. This was all done during spring low tides for a long list of 55 non-native species, of which 41 were invertebrates and 14 were algae. It was really important that we did the surveys on spring low tides to ensure that we had the maximum chance of observing non-native species, as we know they tend to favour the lower tide line. A detailed protocol has been produced detailing the method that was used out on the shore, and this can be shared with anyone if required. Please just contact me. All of the sites, as I've said before, were chosen to be within the boundary of a Solent MPA, and this was vital because we needed to be within the boundary of that MPA to ensure that we could use the data to inform the condition of the site. And a comparable list of native species that were representative of the Solent was also recorded as a means of confirming whether the absence of a species was because the substrate or conditions weren't suitable for the species, rather than because the conditions were not suitable. So, for example, if a native solitary sea skirt was recorded, but an equivalent solitary non-native sea skirt was not, we can infer that it is because the non-native has not reached that shore yet, rather than the substrate not being suitable to support solitary sea skirts at all. The substrate each species was found on was also noted so that we can match this with the features of the site to inform condition. So what did we find? Firstly, there were 35 different non-native species from the target list of 55 found on shores. The 10 most frequent of these are listed on the slide. Out of these 35, 13 are not believed to have been recorded on shores in the Solent before. On average, a shore had 10.6 non-native species, with the maximum number on any one shore being 21. And this data combined together really provided us the first semi-quantitative data of non-native species and confirmed that non-native species were a threat to condition of the Solent. To provide a bit more data on this, we can start to look at a mixed sediment shore, because this is where that trend was really highlighted. So image A shows a typical Solent shore consisting of finer sands and muds that's overlain with cobbles, gravel, shell, and then obviously other bits of debris. And when you look across a shore like this, it can appear at first glance to be quite barren. However, when you start to do that proper focus search of the harder substrate, you start to find it's full of life. Image B is a non-native species called the San Diego sea squirt, or Botuloides diogensis. And this was just found on the bottom of a stone. Image C is the orange-tipped sea squirt, Corella eumyota, again just on the bottom of a stone. And then images D and E show devil's tongueweed, Gratulopia turatura, and wireweed, Sargassum muticum. Both of these species were attached to a small stone that had become buried by the sand and mud and then overlaid. So the algae itself was attached to something buried underneath the sediment. But in all of these examples, the non-native species are occupying space that would otherwise be occupied by a native species. And this trend was consistent for all of the mixed sediment shores in the Solent, mixed sediment being a feature of the SAC. What also became apparent was the influence of man-made structures for increasing the number of non-native species in an area. So the first image on the screen shows another typical mixed sediment Solent shore, just on a slightly sunnier day. But running through the centre of the shore was an old pipeline, which on closer inspection was shown to be teeming with life, including several non-native species. So pulling all of this together, human-generated hard surfaces, and I don't just mean infrastructure when I'm talking about hard surfaces, I'm also talking about debris, so tyres, bits of plastic, anything else that just happens to be washed up, are contributing to the prevalence of non-native species on shores and are likely to be acting as stepping stones to facilitate further spread of non-native species around the Solent, likely giving further evidence for the need to clean up our shores. Several species were recorded in the Solent either for the first time or at least much more commonly than before, and some, now albeit a minority, of these were non-native species regarded as species of concern. For example, the carpet sea squirrel or Didymnum vexillum, which is shown on the slide and is sometimes known as sea vomit. Didymnum vexillum is considered to be a substantial threat to both native biodiversity and also the economy due to its ability to spread and smother any surface, and this includes seagrass and other native fauna. So combined, this data gave us the first comprehensive indication that non-native species were a threat to the condition of the Solent. However, continued monitoring was needed, and I'll talk about some of that continued monitoring later on. The second question, looking at data for the influence of marinas on prevalence of non-native species, we started to see some really interesting trends emerge. And I'll say now that all of this analysis was done by John Bishop at the Marine Biological Association. So perhaps unsurprisingly, marinas had the highest number of non-native species, with an average of 16.7 per marina. The shores nearer to the marina had the second highest number of non-native species, with an average of 12.3, with those far shores only having an average of 8.8 non-native species. So we can see the trend here of the highest number of non-native species in the marinas, the second highest, those shores near to a marina, and then the lowest number on the far shores. And the differences between these were shown to be significant. This starts to indicate that leakage of non-native species from marinas out onto the shores. When we look at the ratio of the number of non-native species recorded to the number of native species on shores, we see that the number of non-native species was higher in the near shore than the far shore. And this reflects the trend on the previous slide, and we believe that this data could be used as a potential indicator of environmental status. Finally, we also looked at site occupancy. So in short, this is just how many sites are actually occupied by each species. And interestingly, the effect of nearshore versus far shore, i.e. the high number of species on the near shore versus the far shore, only applied to non-native species, indicating that the proximity to a marina does not affect the occupancy of shores by native species in the same way that it does for non-native species. So again, we start to wonder whether this reflects the role of typical entry points for non-native species as a source of non-native species colonisation of shores. So again, summarising and bringing all of this together, marinas certainly have the greatest prevalence of non-native species, with shores closest to a marina or harbour having more non-native species than those shores further away. And as I said, this appears to reflect a sort of leakage of non-native species from marinas and harbours onto shores, indicating that they may be influencing presence and colonisation risk of non-native species onto our shores. And this is therefore a potential threat to the good environmental status of our marine protected areas. All of this work combined really does highlight the need for good biosecurity planning, which is why we're obviously all here today. But we also wanted to make sure that the results of this study weren't just a one-off. And therefore, in the late summer of 2022, we went back to 4 of the original clusters, one in West Hampshire, one in Southampton Water, one in East Hampshire and one on the Isle of Wight, deliberately designed to provide good coverage across the entirety of the SAC to repeat the surveys. The work was once again done in partnership with the Marine Biological Association, and we used exactly the same method and the same personnel. And we also ensured that the same person visited the same section of shore or marina that they had done in 2018-19 to provide further consistency with the 2 sets of data. Now, the results of this work are still being written up. However, we have some early interesting findings that I can present today. Firstly, we have the first record on the Isle of Wight of the pom-pom weed, Coelacanthus okamuri, which indicates onward spread of this species across the water from the Solent to the Isle of Wight. We also had the first record for the Solent of the purple fanworm Bispira polyoma, Now, this had previously been recorded in the southwest, but we think these are the first records in the Solent. So this indicates new colonisation into the area. And then we have the pink-hearted hydroid, Ectopleurocrocia. This hadn't been recorded at any of the sites, so any of the 14 marinas or 28 shores in 2018-19, but it was found at one of the marinas in the 2022 surveys, indicating range expansion having previously been recorded on the River Itchen. These surveys also started to highlight some colonisation myths, and of particular concern is the spread of the Pacific oyster Magellana gigas onto shores. So the images on the screen show the same shore. The first image was taken during the original surveys in 2018 and the second image in 2022. Now, whilst the Pacific oyster was present at this site in 2018, it was in much lower numbers. And so we know that this spread has occurred at some point between 2018 and 2022, but we don't know exactly when because we didn't have the boots on the ground. And this is of particular concern because if we're changing the sediment from that typical muddy, sandy habitat into a reef, not only are we losing a feature of our SAC, but we're also losing access to a key prey resource for our SPA birds. So bringing all this together, why are we here? The condition assessment highlighted that potential issue and the need for further study. The EMFF surveys in 2018-19 generated the baseline, and there was also a biosecurity review conducted by APEM, which highlighted that the Solent was a priority area due to the high risk to MPAs and the area being a hotspot for introductions. And that biosecurity review will be covered by APEM in slides later. The EMFF repeat in 2022 built on the evidence base and highlighted the potential adverse condition reasons of non-native species to the Solent Maritime SAC. As I say, the final results of that are pending, but I will share them with everyone once they're ready. And therefore, bringing all of this together, it really highlights the need for biosecurity planning, and these should be good, easy actions that we can all put in place. But also the need for that continued monitoring of non-native species through commission surveys, but also really utilising citizen science, just so that we can continue to track the spread of non-native species across the Solent. Thanks for listening.